Welcome back to the lecture series in animal physiology. So, we have finished 4 weeks. So, today we will be starting our 5th week, and we will be starting this week where we left on the 4th week. So, we discussed about the electrical properties and transmission of signals from the muscle cells to the nerve cells and between nerve cells. And I told you in the very first week that there are 2 ways you can classify cells: excitable cell and non-excitable cells. So, today we will talk about the genesis or the origin of excitability. How a cell become excitable, what we call as excitable cell, and how it varies from a non-excitable cells. So, in order to appreciate what is excitability of a cell or a non-excitability, we probably have to look at the genesis or the evolution of cell itself. It is believed that life has evolved from the water bodies, possibly from the ocean or sea, or this is what at least the current paradigm in evolutionary biology is. And possibly when the first cell must have formed, very first cell on earth, because the earth initially was nothing but very early. It does not have any UV covering. The weather was really horrendous all over the place. There were sparks and lightning, high temperature. To imagine, it must have been a very catastrophic conditions, and under that there were nothing but inorganic elements forming compounds, breaking compounds, and all sorts of elemental zigmo was going on. Probably at some point in a distant past, a framework of certain inorganic atoms took place, which acted as a scaffold to the generation or self assembly of a first enclosed structure, which we today call as cell. This story is mostly based on speculation and based on life forms which evolve in extreme environments, maybe in the sulfur spring or hydrothermal vents, or at a very high altitude with very low oxygen tension and very low temperature, or even people try to mimic the environmental constraints of which exist in Mars. They call it Martian environment or Martian biology. So, we really do not know how possibly the first cell has arisen, but what we can conclude from looking at the present day cell is there are a few features which possibly during 1800, it kind of spread around 1800, the cellular theory was formulated by Rudolf Virchow and all other people, where some of the theories which you must have studied in your school, omne cellulae cellulae, a cell arising from the pre-existing cell, which essentially means a cell can create its own replica from the pre-existing one. So, to start off with, let us formalize whatever we are talking. So, this is we are into week 5, lecture 1, W5/L1. This is where we are. So, what we are dealing now is origin of excitability in cell. And before that, we are talking about prior to that, origin of biological cell. It could be anything from bacteria to mammalian cell to plant cell to fungus, anything. But the very first cell which decides that it is one entity which follows the thumb rule, cell arising from pre-existing cells. Okay. So, I told you that the very first, so if we whenever we draw a cell, we draw it like this. Of course, now we have already introduced the neurons which have a different kind of architecture. So, this is the mitochondria, cell membrane, endoplasmic reticulum, okay. Then you have Golgi apparatus and like so on and so forth. It is a very complex structure. So, the very first thing which may have happened is the formation of the cell membrane. That whole assembly of phospholipid bilayer which made up the cell. How it formed that structure is a completely unknown thing, but it is believed that the very first molecules, whether it is DNA or whether it is a series of proteins or even as a matter of fact the lipids, they all needed a framework to form. Framework or you can call it a scaffold framework or some kind of a template, which is the right answer, or a molecular template or an atomic template. It is this initial template, we have no idea how this template was formed, what made these molecules to self assemble. So, it is something like this. What I mean to say is that, say for example, you must have, all of you must have seen templates. Suppose this is a template. Okay. So, if you put some jelly like stuff into it which is fluid, then this new stuff will take a shape like this, right. Now, as soon as you remove the template, so what you are going to get is something like this. One second. Okay. This is what you are going to get. So, your template was the black one, this one was your template, and on top of the template this is what has formed. So, exactly same way there must have been certain atomic templates on which these structures may have formed, but then how in the process they develop the machinery to store this information, that they can do this act n number of times, we do not know. Where possibly DNA learned that it has to store the genetic information, we do not know. These are unanswered question of chemical evolution, because prior to the biological evolution, there must have happened certain chemical evolution which led to the formation of these kind of templates. That part is, we all are in dark. We just have series of speculation to tell what may have happened. Possibly these things must have happened, at least this is the timeline of past we are trying to recreate, 4 billion years, or even maybe more. It could be even 4 to 5 or 6, whatever. We are trying to recreate a history of atoms and molecules which are 4 to 5 billion years old. We do not know really, but what we know now, in between there is a complete dark corridor through which we have traveled, for which it is really tough unless we could go back in time to figure that out. But what we know is this: when we talk about a cell like this, we realized the cell, if assuming that this cell has evolved, or this kind of cells, and of course, if it is a plant cell, then there you will have another additional membrane which will be the cell wall. Okay. Though I am not getting into those finer details at this stage. So, say for example, if it has evolved in the very salty water of ocean, salty water of ocean or sea, where the salt concentration is very, very high, so high that nothing really could grow in that direct contact with it. So, it possibly learnt a way to create an environment around itself in a way by forming a capsule or a structure what we call a cell, where this concentration of sodium and chloride out here is fairly low. So, in order to do it, it had to create a boundary, what in modern days term, which is explained by fluid mosaic model, Dixon-Joly followed by fluid mosaic model during 1960s, what we call as cell membrane, where sodium inside it, sodium and chloride concentration is fairly low, and it has a high concentration of potassium as compared to outside. Potassium, one second, potassium is low outside, and calcium contrary is high, but there is no free flowing calcium because it has one organelle called SR, just like ER stand for endoplasmic reticulum, SR stands for sarcoplasmic reticulum, sarcoplasmic reticulum, SR. So, the calcium is mostly remain in a stored chelated form in the sarcoplasmic reticulum. So, the very first signature for a cell to evolve was the formation of a membrane. How that membrane was formed, we do not know. We have no clue about it. Now, once this membrane possibly was formed, the next criteria for this membrane was, so now we have enclosed, say for example, the first cell which is formed, it has kind of some way or other managed to have a lower sodium concentration inside it, but then there will be always diffusion, right. The diffusion pressure will ensure that there will be motion of sodium and which will balance it out. But it is at that time there is another unknown story evolved. It develops structures what in modern days biology we call them as ion channels, and it develops something even more interesting what we call ion pumps, or you can just call it pumps. So, in other words, it form a house with a pump. If by chance from surrounding water gushes in, it has a pump to throw away that water. It is exactly the similar analogy happened here. But now here is again something unknown. How this evolved? Who told that to evolve like that? Who decided the structure of the channels will be like that? Well, we do not know these answers, but what we know today, now coming to with giving you this 4.5 billion in last 10 minutes, what we know today is our cells have, say for example, if this is the cell, and now I am talking about the animal cells only. So, I am not taking into account the cell wall, I am just talking about the cell membrane. If this is the cell membrane, the first level of self assembly forming a defined definitive structure by the self assembly of the advanced organic molecules, it is like this, right. All of you have studied it at some point or other. It is a lipid bilayer having the polar head groups of lipids, polar head groups, and hydrophobic tail. So, each lipid molecule is something like this. This is the polar head group and this is the hydrophobic tail. And when they self assemble, in order to minimize its surface energy, all the polar head groups faces either outside or inside. So, all these are polar head groups. They can interact with water out here, the polar head groups, okay, P, P, whereas this part, what you see, what I am shading in red now, this part is extremely water hating. In other words, it is a hydrophobic region. Now, on this membrane are embedded certain unique structures like this. Some of them are pumps, cation channels, anion channels. Okay. Major channels which will be concerning here will be sodium channel, potassium channel, and as we will go further, while we will talk about disease like cystic fibrosis and everything, we will talk about chloride channels. There are water channels which are called aquaporins. Okay. Then we have these pumps. At this point, we will be dealing, of course, as we will move to kidney and other places, kidney, digestive system, we will talk about other kind of pumps. We will be talking about sodium, potassium, ATPase pumps. So, earlier to this, I told you that inside the cell, sodium is low as compared to outside. As a matter of fact, our whole body, if we consider our body like this, these are the cells which are forming the structure. between the cells there is extracellular fluid. So, the extracellular fluid is here where I am shading with blue, right. This region of extracellular fluid, if you have a siphon or if you have some way to remove the extracellular fluid, if you have some kind of some needle or something, right, and you can pull out the extracellular fluid like this, then you will observe the concentration of extracellular fluid is almost similar to that of the sea water. It has very high sodium, very high chloride, anion and the cations, whereas as compared to the sodium inside, you will see sodium is low, but potassium is fairly high. Okay. So, this is extracellular. So, all our cells in the body are bathed. So, if you look here, so all the cells which are forming this structure of my hand or this, they are all bathed in high sodium concentration, which is the reason why sometime we are given saline or something, we lose body fluid or something. So, all our cells are bathing in a similar condition as that of seawater, but inside the cell the story is different. Okay? Inside the sodium is low. That brings us to a very interesting paradigm. What is that paradigm? So, inside I told you the sodium is low and outside I told you sodium is high. So, automatically by the diffusion process, this outside sodium should try to move inside like this. And similarly, inside the potassium is very high. I am not giving the values, I will give you in the next class purposefully, but at this point try to understand the basics. Okay? By the logic, this potassium which is inside should prefer to come outside, because by the gradient potassium is at a much more higher gradient potential. These circles tells you who is having higher concentration, right. But having said this, this brings us to another aspect of it. I talked to you about the chemical potential, but there is a charge aspects to it. So, before I get to the charge aspect, there is one interesting thing which I needed to point out. If you take an electrode and place an electrode inside the cell, and there is an electrode you left outside very close to the cell of course, and you take a voltmeter and measure the voltage across the membrane. In other words, you are measuring the voltage across the cell membrane. So, inside the cell membrane versus outside the cell membrane. It has been observed for all the cells of your body. I told you that all your cells are bathing in extracellular fluid like this, right. Now, out here what I am asking you, you put an electrode here, you place an electrode here and you measure the voltage across it. You will observe with respect to the inside the cell, with respect to inside the cell, outside is more positive. With respect to inside, outside is more positive. So, it means out here what will you observe is inside with respect to the outside, the voltage is inside is around minus 70 to minus 80 millivolt. There are few cells which all the ways goes up to minus 40. I will come later about those. At this point just remember, but this is not absolute fact that it is minus 70 to minus 80. There are cells which are at minus 40 also, but this is what was observed. All the cells, irrespective of the fact whether they are excitable cells or they are non-excitable cells, okay? Whether they are excitable or non-excitable, all these cells maintain a cell membrane voltage, okay, across inside to outside at minus 70 to minus 80, barring aside few cells which are called the pacemaker cells in the cardiac system, as well as some of them are sitting in other places of the nervous system, which sits at minus 40 millivolt, irrespective of anything. But then what is this excitability, non-excitability? Now, while closing on the class today, I will give you one small clue. Excitable cells are the ones. Excitable cells are the ones. So, this is the one I am talking about. Okay? Excitable cells are the ones where this from this value all of a sudden it fluctuates like this. From minus 70 to minus 80 millivolt, it fluctuates to positive more than 0 and comes back very transiently, as if all of a sudden there is a breakdown in the cell voltage. There is a load shedding, there is a breakdown. All of a sudden this whole across this membrane, this becomes 0. From minus 70 millivolt, it becomes 0 and little more than 0, even much more positive, and it comes back. So, what you see on a scale, if you plot the voltage on the y axis and the time on the x axis, and if the cell is sitting at minus 80 millivolt, all of a sudden you see something like this, and this is the classic characteristics of an excitable cell of your body. So, it means this feature is shown by all the neurons, all the muscle tissue, all the cardiac tissue, all the smooth muscle tissue, and the neuroendocrine cells. This is the signature, and that distinguishes excitable cell from the non-excitable cell. Non-excitable cell remain the status quo of maintaining minus 70 to minus 80 all throughout their existence. They do not change unless otherwise. We will come later into that, how a non-excitable cell can become excitable, what is the genesis, how it can become. And actually that is where lies some of our very fundamental questions about stem cell biology, the way the field is moving, especially neural engineering and muscle tissue engineering and all that stuff. But next question is that if excitable cells are the ones where all of a sudden it shoots a spike, that the membrane potential, what we call as minus 7 to minus 80 is the membrane potential. It is the membrane, it is the potential across the membrane. If that changes so sharply, how or what are the players, not how comes next, what are the players who can make that happen and under what conditions such things happen. Okay. So, I will close in here. So, we are closing on the very first class of the 5th week. So, in the next class we will talk about how this spiking happens, and that will introduce us formally to the world of action potentials, okay, because this action of coming from minus 80 going all the way to overshoot 0 and comes back is called an action potential. And that will take us to the story of Hodgkin, Huxley, Cole and all those other people, even Sherrington, the discoverer of synapses and all that. Okay? Thank you.